Layman seminar today, and I want to start out and thank the people who are responsible for suggesting the speakers and putting the program together so that we can bring this to you today. The Office of Research Administration, of course, the UCLA Development Office, the Research Administrators Campus Committee, who I believe is the selection group for the speakers, the team in the Dean's Office, specifically Dion and Rosalie, and to all the research administrators throughout UCLA. You are the people that make research possible, and we're very much indebted to you and very grateful to you. And we think it's great that you're interested to come and see some of the work that you're supporting. So I'm Judy Gasson. I'm the director of the Cancer Center and the Senior Associate Dean for Research in the David Geffen School of Medicine. And I really want to welcome Dr. Sam here, who's going to be our speaker. He said everyone calls him Dr. Sam unless they're angry with him, so I'm just going to go with that. Sam is truly a California man. He was born in Southern California. He went to Berkeley for his undergraduate work and actually came to UCLA for his Ph.D. Then he started his northern migration again, went to Stanford as a postdoc, and the University of Washington as a senior fellow. And he must have gotten a whole good look at that Seattle weather up there, because he returned to Southern California, never to leave again after that. He started out as a professor at USC, and he's now associate professor here at UCLA in the Stein Institute. What he works on is absolutely fascinating. He works on the mechanism by which the retina works. You all know the retina is in the back of your eyeball, and that's how we're able to see. And he studies both the molecular and cellular activities that allow us to see, but then links that to how human behavior is changed. I think this is fascinating. I'm really looking forward to hearing it. So please join me in welcoming Dr. Sam.
Let me turn this. Can you hear me? Does this work? Did you all hear me? Well, great. Well, first I'd like to thank the Research Administrators Committee for allowing me to come and tell you about our research. It's really quite a privilege for me to be here today. The one thing that I found perhaps just a little bit troubling about this whole process is the fact that my picture has been plastered on every corridor intersection of every floor of CHS. And, normally, so I've been ducking my head and walking around the building, But I came across one particular sign just yesterday on my way back from the Jules Stein Eye Institute, the Life Science Building, which is this one. So I took this back home to my wife today, thinking I could make her jealous, and she had very much the same reaction that you all did. And I could have an admirer, maybe. But in any case, what I'd like to tell you a little bit about today is I'd like to tell you about the work that I've been involved in over the last 10 years since I set up my independent lab at USC, and now here since October. I've been interested in one facet of the work that we've been working on is relating to establishing how we see near our absolute visual threshold. Now, absolute visual threshold is a term that I'm going to use repeatedly, and what this refers to is the minimum amount of light that we require in order to perceive that we have seen light. So if you were to take a person and keep them in a dark room for an hour, 2 hours, 3 hours, and test them, what you would find is that absolute visual threshold is actually a very low intensity of light that's going to be much lower than what you expect it to be. And you'll see what I mean as I go forward. So across vertebrate species, as it turns out, the fundamental architecture of the eye is totally conserved. So here's some examples of all the different classes of vertebrates, including mammals. Should we turn this off? The front lights off, perhaps? Is there a way to do this? Lighting. Stage lights. How's that? Okay. Chris Evans calls this the IQ test, which is funny, if you can actually work that thing. But in any case, across all vertebrate species, the fundamental architecture of the eye is almost identical. Okay. So if we look at mammalian species all the way from monkeys and cats, rabbits, rats, and mice, they have the same fundamental eye plan, the same cell types as the retinas or the eyes that we see in birds, reptiles, amphibians, and even fish. So given that we're interested in how people see as a way of gaining a window into our visual perception, let me give you this example of a couple of mammals, primates in particular. Quite cute primates, if I do say so myself. In fact, this one is in her natural habitat. But if we were to look at the eye and magnify out the eye, what we'd see is this fundamental architecture that I described. And you can think of an eye very much like one of these old SLR cameras and one of these new digital SLR cameras. The front half of the camera is all about focusing and light, right? So the front half of our eye has a cornea and a lens that allows us to bend light and focus it, and their iris, which constricts to make a pupil that limits the amount of light entry. That can be analogous to an f-stop of the camera. And so the front elements of the eye take light that's reflected off of objects in the environment and focuses it onto the back surface where the film is. Now, unlike these old cameras that have a static film, our retinas are very much like these new digital cameras with these CMOS sensors. They do all these calculations before the signal even gets to the brain, including gain control, some simple image processing, etc. And this is actually what the retina does as well. But the retina, of course, is a biological tissue which consists of layers of distinct cell classes. This is a schematic of the retina to the right here. And what happens is that light that enters our eye essentially passes through the retina, which is a largely transparent structure, and it strikes the outer segments here of our photoreceptor cells. You might have heard of the rods and the cones. These two kinds of photoreceptors operate at very different light intensities. Rods are exquisitely sensitive to light and are responsible for our night vision. But cones are much less sensitive to light and are capable of supporting vision, for instance, in this environment. And cones come in many varieties. Unlike the one type of rod in all vertebrate species, there are 3 kinds of cones in primates and 2 in most other mammalian species. And these absorb best in the red, the green, and the blue. And it's their relative activity as processed by the retinal circuitry that tells us about color, about motion, about more complicated features of visual space. Now, this outer segment in here is the light-sensitive part of the photoreceptor cell. It contains the visual pigment, which is the molecule that absorbs photons of light. And it also contains all of the signal transduction machinery that allows the absorbed photon of light, or the photon being the smallest particle of light, to be converted into a change in the cell's membrane voltage. So the cell's membrane voltage, in turn, controls from its terminal end, its output end, the release of a neurotransmitter, which in this case is the molecule glutamate. So glutamate is released from these photoreceptor terminals, from either rod or cone synaptic terminals, onto the intermediary class of bipolars in the retina called bipolar cells. Bipolar cells, in turn, when they receive glutamate, it changes their membrane voltage, and they in turn release neurotransmitters onto the ganglion cells, which are the output cells of the retina. Now, the ganglion cells in turn send axons that go to the middle of our brain, the visual thalamus, and eventually project back to the visual cortex. Now, the immense importance of vision to us as a species can be seen just by looking at our visual cortex. And I should be careful what I say here because James Beasley is sitting right here. But the importance to vision as us as a species is reflected in the fact that perhaps 1/3 to 1/2 of our cortex is dedicated to processing information that comes through our eyes. So the 100 million photoreceptors that are present in each eye are compressed to about 1 million ganglion cell axons that go to the middle of the brain. So there's a tremendous opportunity to do pre-processing in the eye before the signal then gets sent to more central structures. Now, this is a 2-dimensional structure right here, but in reality, if we look in 3 dimensions, this is what an electron micrograph of a frog retina looks like. Okay? So you can see here the rod outer segments are these rod-like structures here. Right? They're very long. They're optimized for catching photons of light. Interdigitated between them, you can see these little cone outer segments. Now, just to give you a sense of scale, in this direction, there's about 20 rods. In this direction, there's about 20 rods. So this pool has somewhere in the neighborhood of 400 rod photoreceptors in it. This is going to become relevant as I go forward in my talk. But what I'd like you to take away from this slide is something very simple. Light from the environment is reflected off of objects. It's focused by the front half of the eye. It's then absorbed by photoreceptor cells in the back half of the eye in these outer segments. Their activity changes. They relay those signals to bipolar cells. These bipolar cells then send them to ganglion cells, which then get projected to more central structures to allow our highly robust and complicated vision. Okay? Now, let me turn back now to the research problem that I've been involved in for the last 10 years, is how do we see near absolute visual threshold? Well, this now goes all the way back to experiments in the 1930s. Long time ago. This was right around the time that we, as a people, began to become very good at manipulating and measuring light intensity. So in this seminal work from Selig Hecht in the 30s that was published in 1942, he and his colleagues set out to establish exactly how many photons of light we need to absorb in order to have the perception that there was indeed a light stimulus. And so they built a very simple optical bench, right? It consists of a lens, a set of filters to control the light intensity. This M1 and M2 are something called a monochromator to make sure that the light is green. And the exposure to the eye is controlled by a shutter, so you can have a very brief flash of light. The subject whose head is fixed is here. This is just a schematic of the eye. And they're fixating at a focal point. And so this spot of light produced by this lamp subtends about 500 rods on the periphery of the eye. Now, the reason it's on the periphery of the eye is probably for a reason that you already anecdotally appreciate. If you've ever been camping and you look up at the night sky, sometimes in order to see a star, you have to look slightly away from it. And the reason is that the center of our visual field, where we're fixating, goes to a part of the retina called the fovea, which is all cone photoreceptors. And indeed, rod concentrations start to increase as we go peripherally from the fovea. So by looking a little bit to the side, you're focusing the spot on the part of the retina that has the most rods. Okay? So they basically put themselves, Hecht, Schlar, and Perrin, into this apparatus. This is Selig Hecht, Simon Schlar, Maurice Perrin. They dark adapted themselves, which means that they put themselves into a dark environment for a long period of time so that the visual system was maximally sensitive. And then they delivered these various strengths of light, and they reported whether they saw it or didn't see it. Okay? And they built a very simple model. Given that light follows a statistical framework, Poisson statistics, they assumed that the threshold number of photons would be some number or greater of photons. And if you were to look at what's called a cumulative Poisson distribution, is that depending on that threshold number, the steepness of this curve becomes greater and greater. The greater number of threshold photons, the steeper the curve. And so what they did was they took their behavioral data and they fit it based on the steepness with one of these curves. And indeed what they found is, based on this spot of about 500 photoreceptor cells, that each of them, their threshold was somewhere in the neighborhood of 5 to 7 absorbed photons. Okay? Now, given 500 photoreceptor cells and 5 to 7 absorbed photons, statistically, it's very unlikely that a single rod would have absorbed 2 photons. So the likelihood is that the vast majority of visual processing done at this light level must be due to single photons that are absorbed by individual rod photoreceptors. And this was the conclusion that they drew from this work. And in fact, much behavioral evidence from this time suggests that it's not just a few photons absorbed among 500 rods. It might even be a single photon absorbed per 10,000 rods. This is quite exquisite sensitivity. Now, the discovery that individual rod photoreceptors can reliably signal the absorption of single photons was later shown by Dennis Baylor and his colleagues Trevor Lamb and King-Wai Yao. As it turns out, I was Dennis Baylor's last postdoc, which gave me the unfortunate nickname in the field of being the straw. But I assure you that I wasn't the cause of his retirement. At least he never actually told me that. But in any case, What Baylor, Lamb, and Yao did was they developed a method of recording from cells using a polished glass electrode, where they were able to suck in the outer segment of, in this case, a toad rod, okay? And then measure the current that flows across it. So here is a sample of what one of those records looks like, where I'm going to plot the current here. You note that this is a picoamp, okay? A pico is a 10 to the minus 12. It's a quadrillionth of an amp, okay? So these are very, very small signals. And what they found, this picoamps against time. This is 30-second time base. And each one of these ticks represents the timing of a flash of light that's very dim, so that it statistically either produces a photon absorption or it doesn't. And what Baylor, Lamb, and Yao found was that as you looked across the record, let's take this middle panel here just as an example, you get these very discrete responses that have a stereotyped time course and shape. You know, sometimes you get failures, sometimes you get singles, sometimes you get something that looks, you know, about twice the size. This was the first evidence that showed that rod photoreceptors are indeed capable of signaling the absorption of 0, 1, or 2 photons. So, along with the previous work, that the human visual system is sensitive to just few photons among thousands of rods, and the fact that rods indeed can signal single photon absorptions, this led to the greater appreciation that the single photon response in individual rods is a very important part of our vision processing. Now, I should admit, I just want to make an extra strong point about this. The ability to resolve the absorption of a single photon of light is really quite remarkable, because in this case, we're limited only by the physics of light. We're limited solely by the ability to divide light into its smallest particles. Right? And this is not just a photoreceptor party trick, okay? Across all of our senses, they all share very similar sensitivity. If you were to look at an olfactory receptor has been shown to be sensitive to the binding of single odorant molecules. Or hair cells in our cochlea are sensitive to vibrations that are on the order of the Brownian motion that they normally experience based on temperature. So the fact that sensory receptors reach the limits imposed by the physics of the stimulus is a very generalizable concept. And so anything that we find in the visual system for how these signals are processed is likely to be reflected in the way other sensory systems process this information. So what does the single photon response mean with respect to our ability to see? Well, if we consider the full range of light intensities over which the visual system works, the darkest of nights to the brightest of days, it encompasses about 10 to the 12th fold, or about a quadrillion fold in light intensity. That's a huge range. Okay? If you took a photon absorbed per 10,000 rods near our absolute threshold, go to a photon absorbed per 1,000, 100, 10 photon per rod, the lower 4 orders of magnitude, or 10,000 fold in light intensity of our visual system, or a third of our total dynamic range of our visual system, is encoded by single photon responses in individual rods. Now, this has some very important implications for function and disease. The first is that being able to resolve the single photon response becomes important for vision in this range. And any manipulation, whether based on disease or otherwise, that deteriorates your ability to resolve a single photon response amid the noise in the receptor cells will always lead to night blindness. Okay? So there's a very real functional correlate to the loss of your ability to resolve single photon responses. Let me go a step forward. As we age, our ability to achieve sensitivity where we can see single photon responses, this is a process called dark adaptation, becomes impaired. And so as we age, our ability to put ourselves into a state where you can photon count becomes slowed as well. And this, phenomenologically, is the reason why, as we age, our night vision becomes progressively less and why driving becomes more difficult, etc. Okay, so single photon responses, not just a party trick, actually incredibly important over a wide range of light intensities, the bottom 4 orders of magnitude. And so what we've been interested in is trying to understand how the single photon response traverses the retinal circuitry and what it means for behavior. And so what we've done is we've looked at the mouse retina. Now, as I mentioned to you, across all vertebrate retinas, they share a common architecture and they share all the common cell types. But mammals even share something more. Mammals all share common circuitry. And what I mean by that is that they have the same cell types and they're connected to the same cell types within the circuit. And in particular, in the mammalian retina, there's one pathway that conveys the signals of rods to the retinal ganglion cells. It's called the Rod Bipolar Pathway. And this is a pathway that pools thousands and thousands of rods in a series of steps. About 100 rods are pooled onto a rod bipolar cell. This is a specific kind of bipolar cell that contacts rod photoreceptors. And then these signals are fed. 25 rod bipolar cells feed an intermediary neuron called an A2 amacrine cell, tens of which feed the outputs of cone bipolar cells, such that ganglion cells that look up into this circuitry are seeing thousands of rods. Well, the fact that there's this defined circuitry gives us a very unique window into this problem of how single photon responses are relayed through the circuitry, because what we can do now is, using physiological techniques, we can go through and we can record from every single cell type. Okay? So I showed you what the suction electrodes look like from Dennis Baylor's experiment. So using suction electrodes, we can now record the membrane current from the outer segment of rods, right, as shown here. This, just to describe this for you, this is what we call a flash family. And a flash family is essentially a plot of the membrane current against time. And this arrow represents the timing of a very brief flash of light. And that flash of light varies between something that's incredibly dim and doesn't really elicit much of a response, all the way to those that elicit a maximum or saturating response. So with very high resolution, we can characterize the properties of the signals and actually also the noise of photoreceptor cells. The noise will become important as we go further. We can do the same thing going through every cell in the circuitry. From bipolar cells, we can take a dark adapted tissue. We can make slices of it to preserve the circuitry. And we can record from them using something called the patch clamp technique, where we take a tiny glass electrode and make a high resistance seal against the cell. And then we can measure a very similar flash family from a bipolar cell or from a retinal ganglion cell. So what we have here is we have the opportunity to look with very high precision quantitatively at how the property of signals and noise vary as the single photon response traverses the circuitry. Now why is this important? This is important because we can now compare this with visual behavior. So what we've done is we've employed a water maze. And the water maze allows the mouse to try using visual cues to find the black target against a white maze. Now the water is cold, which motivates the mouse to try to swim to the platform and crawl out. Although I've not explicitly checked, I'm sure there's some shrinkage. But we can use the amount of time on average that it takes for the mouse to find the platform as essentially a proxy for how well it can see. So this was a case of how it's done in bright light. Let me go around here and show you what this looks like in the dark. So this is now in total darkness using infrared light and an infrared camera. So you can see right here that the platform's right here, but the mouse can't see it. And it will swim randomly looking for it to climb out. This might seem very cruel, but we don't let the mice go beyond 30 seconds or so just to take them out of the water so they don't become hypothermic. But what we can do is we can start to lower the light intensity from light all to darkness where they can't see. And what we can then generate is something that looks very much like what Heckschler and Perrin saw. We can plot an escape time, or this is equivalent to the percent correct, against the background light intensity. And we can see the same sigmoidal relationship. And in fact, if you took the inflection point of this curve and did a calculation, it would work out to be about a photon absorbed per 2,500 rods per rod integration time. So these mice are behaving at a light level that's very similar, of the same order of magnitude as behavioral threshold in humans. And to go a step further, we know what the circuitry is that's carrying these signals to higher visual centers. So this puts us in a very unique opportunity to study how the properties of signals and circuits within the retina directly cause visual perception. Okay? And it's for this reason I find this to be a very powerful methodology. This is a methodology that it's not universal across all all preparations. We have a degree of control of this system that's really unprecedented compared to people who are working on hippocampus or even working on cortex. Sorry about that, James. So with that in mind, what I'd like to do is I'd like to take you through some experiments now that we've done to try to identify some of the mechanisms that are involved in helping set our visual threshold. So a photon absorbed per 10,000 rods, visual threshold. A photon absorbed in a single rod generates a change in the membrane voltage of the rod of something like 1.5 millivolts. It's very small. If you consider that in lieu of the fact that near threshold, only one rod is absorbing a photon, but 9,999 others are just generating electrical noise, you have a bit of the problem. You can't just linearly add together what 10,000 cells are doing. Otherwise, you're going to lose your tiny little single photon response from one rod in lieu of all the noise that's present in all of the other rods. Okay? Now, it's appreciated throughout brain processing that we do process very sparse signals. So has anybody here heard this anecdote before that we only use 3% of our brain? Have you guys heard this before? Well, the reality is that's not actually true in the way that we think about it. We actually use all of our brain. It's only on 3% at a time, okay? There's a big difference there, right? And what that means is that in any given neuron in our brain, only a small fraction of its inputs are active at any given time. And so this idea that we process sparsely information that's presented is very generalizable. And in fact, this is also true in this case. So the analogy that I like to give here is this. Let's say that you're the UCLA quarterback, Brett Hundley, okay? And you're in the middle of the Rose Bowl, and you look into the stands, and you see 10,000 screaming fans. But your mom is there too. Go mom, okay? So if you wanted to hear what your mom was saying in lieu of what all the screaming fans were doing, you couldn't just hold up a microphone pointed to everybody and add up what they're saying. Because there's no way you would hear what your mom was saying compared to what 9,999 other people are saying. However, perhaps there's something special about what mom's saying. Perhaps she's yelling a little louder than everybody else. Well, the way to pick out mom from this group is to be able to take your microphone and go person by person and measure the amount of sound that they're generating. And you would keep the sound if it increased past some threshold, and you would throw away the sound if it didn't meet this criterion. And as it turns out, this kind of thresholding is a very general phenomenon of signal processing in the nervous system. And in fact, this is happening at the very first synapse between rods and the rod bipolar cells. So what do I mean by this? So once again, this is a photocurrent from a rod photoreceptor. This is recorded with a suction electrode from the outer segment. I'm plotting the current again. You can see this is very small, like 2 picoamps is this bar length, as a function of time. Each one of these upward triangles represents a flash of light that on average generates less than 1 photon absorbed per trial. So what that means is that statistically you'll get some trials where there's no photons absorbed, some that are 1 photon, some that are 2 photons. This is what the average single photon response looks like. But if you were to look at a trial-by-trial basis from a single rod, it's kind of hard to tell in some cases which are single photon responses and which aren't. Right? Is this a photon response or is it just noise? This probably looks like a photon response. Yeah, this is probably a double photon response. This is twice as big as the 1 picoamp of this response. So if you were to deliver 1,600 or so flashes to this rod photoreceptor, and then plot the amplitudes of all these flashes correlated against this single photon response, you get a histogram that looks like this. So this is the number of events against the amplitude. Okay? So amplitude moves now from this axis to this axis. Around 0 picoamps is our noise, or it's where no photons absorbed. Right? And it has some Gaussian distribution. Then we have, at 1 picoamp, roughly the single photon response that also has a Gaussian distribution, and around 2 is the double photon response. And as it turns out, this curve is beautifully fit as a sum of Gaussian distributions whose weights are determined by the Poisson probability of absorbing a photon. So said another way, the statistics of all this work out perfectly. Okay? Now here's the problem. If you're now a bipolar cell, and you're looking up at one of these rods and you're trying to figure out whether it absorbed a photon or not, you have a problem. And the problem is this. It's all this overlap between the noise centered around 0 picoamps and the signal centered around 1 picoamp. If you have an event in this area, you can't uniquely tell whether this is just a large noise fluctuation or a small single photon response. And this is where thresholding actually helps you out. So if you were to impose some kind of threshold here, I've drawn it horizontally here and vertically here to represent the same threshold. You can imagine that if you thresholded it here and you kept everything that exceeded the threshold, and you threw away everything that didn't, you might do a little bit better, right? One way of saying that is if we position this optimally between these two distributions and we kept everything to the right, we're more likely keeping things that are single photon responses compared to noise. And if we throw away everything to the left, we're more likely throwing away noise compared to single photon responses. So this might be the optimal place for us to separate these two distributions. And indeed, in rod bipolar cells, if you were to look, the situation is actually much more clean. So these are single photon responses from a rod bipolar cell measured with a patch electrode, and these are noise trials. And so now we go from this distribution where the Gaussians are overlapping considerably with each other to one where they are not now. We can much more reliably pick out a single photon response from the noise. Now, where this threshold is positioned actually becomes really important, because if we put it too high, we're going to lose single photon responses. If we put it too low, we might get all the single photon responses, but we're also going to get a lot of noise with it. So there's an optimization problem that the bipolar cells are doing in where to position this threshold in order to maximize your ability to resolve single photon responses compared to the noise. So I worked on this as a postdoc, and what we found is that this is produced in the bipolar cell itself. So at each individual one of these contacts, there's a threshold imposed on each rod, and then the rod bipolar cell then takes all these thresholded outputs and then adds them together. And that's how we get responses that are much more robustly separated between single photon responses and noise. Okay? So what we want to do now is we want to be able to explore exactly how these processes are set with respect to one another. So what I just described for you is that when you took distributions of signals and noise from a photoreceptor, we had to optimally separate them in order for single photon responses to be seen compared to in the rod bipolar cell, compared to the rod. This problem only gets worse and worse as you go deeper in the retina, because as you go deeper in the retina to the ganglion cells, they're pooling more and more rod outputs. So performing this kind of operation as far out as possible makes a lot of sense. Okay? But there's a really unique opportunity here, because there's this threshold that's separating signals from noise between rods and rod bipolar cells. We can now begin to use tools at our disposal to understand a little bit more about how this threshold works. Now, one tool that we've used extensively is transgenic mice, and you probably have seen tons of proposals that have transgenic mice in them. So what transgenic mice are mice where some element of the genome has been changed, right? And that in turn will lead to the change in the expression of certain proteins that are involved in processes such as the generation of the light response. Let me show you what I mean by that. So this is a wild-type rod or a normal rod from a mouse. This is, again, a suction electrode recorded from the outer segment where I'm plotting the current as a function of time. And we're going to deliver a flash right here. And, of course, the flash starts out to be very dim, and it becomes brighter and brighter until the response becomes maximal. We are going to use now a particular kind of mouse that's missing a photoreceptor protein that's present in the outer segment called GCAPS. Okay? You don't really need to know what GCAPS is, except that it's responsible for providing the feedback that shuts off the light response. Okay? So if you were to take the single photon response of a wild-type rod, it's right here. The single photon response of the GCAP knockout is about 3.5 times bigger and much slower. And I'm not showing this here, but there's also extra noise. So the question is how are the signals and noise increased in this instance? So this is the same data I showed you previously. This is a wild-type rod where we're plotting individual traces. And we have histograms that have this problem of having overlap between the noise centered around 0 and the signal centered around 1. Now, in the GCAP knockouts, remarkably, the single photon response becomes much more distinguishable from the background noise, as we can see here on individual traces. And in fact, if we looked at the histogram of responses here, that's borne out here as well, right? You can much more reliably separate the peak that results from single photon responses compared to traces where there's no photon absorbed. Now, if we make a very simple calculation of what we call the signal-to-noise ratio, the difference between these two is a signal-to-noise ratio increase of a factor of about 1.5 in GCAP knockout rods compared to wild-type rods. Now, we found this result to be very interesting. And the reason is this. Across all of our sensory systems, it's widely held that the signal-to-noise ratio of the sensory receptor cell, in our case the rod photoreceptor, controls ultimately our behavioral sensitivity. That's believed to be true for the olfaction system, for the auditory system, etc. And to some degree, that has to be true because we're limited fundamentally by the quality of the signals that are generated by the receptor cells. So in this case, if we have mice that have a higher signal-to-noise ratio of the single photon response, maybe their visual threshold is lower, right? That's the natural extension of this. They can see at lower light intensities than normal mice. So we looked at this. We put these mice in the water maze, and we tested their ability to escape. And what we found actually was just the opposite. Okay? So I'm just showing 3 light intensities here. Each one of these circles represents 1 mouse compared to 8 knockout mice. These are all litter mates from the same mother and father. So they're age matched, they're strain matched. And as it turns out, in bright light, they both find the target and escape pretty quickly with a relatively short amount of time. And in darkness, they both equally can't find the target and escape. But in this transition zone from being able not to see the target to seeing the target, there's a distinct difference in classes. The wild-type animals are shown here, and the GCAP knockout animals' vision is reasonably impaired under these circumstances. So under circumstances where the wild-type or normal mice can escape from the maze, kind of, it looks like the GCAP knockout mice can't at all. Okay? This is a bit of a paradox. How is it that you can have sensory receptors that have a higher signal-to-noise ratio but have worse behavioral sensitivity? This doesn't make any sense at all. Unless, for some reason, there's other processes in the retina that are deteriorating your ability to resolve single-photon responses in the GCAP knockout mice. So we looked downstream. So these are the wild-type Rod bipolar cells. Again, we're recording with patch electrodes. This is a current as a function of time. Here are the individual traces where you can see clear single-photon responses and clear zero-photon responses. When you do this same experiment in the GCAP knockout, you see that there's a tremendous amount more noise than there used to be. Okay? So as I mentioned before, the signals in the GCAP knockout rods are bigger, but the noise is actually bigger as well. But the signals outweigh the noise enough that the overall signal-to-noise ratio is higher. But interestingly, there's a lot of noise that appears to show up in the GCAP knockouts mice that occludes your ability to see the single photon response. You can see these in these 2 distributions. While this distribution is very discrete, where we can pick out single photon responses compared to dark noise, now they're a little bit blurred with respect to each other. And this looks like it impairs the ability of the mouse to be able to escape from the water maze. This is a very subtle change in signal-to-noise ratio. So let me sum this up for you. Rod photoreceptors that have signal-to-noise ratio, that's modest. On average, you can pick out a single photon response from the dark noise, but there's considerable overlap in these distributions. If we apply the threshold here, as I described it before, we can clean these up, and so now we can have a very distinct single photon response from the noise. We take the GCAP knockout rods that start off with a better signal-to-noise ratio, but somewhere in the process of applying this threshold, we don't see much change in the ability to discriminate single photon responses from noise. So it would seem that under these conditions that the nonlinear threshold is not set optimally with respect to the distributions of signals and noise in the GCAP knockout rods, like it is under normal conditions. Okay? So let's consider a very simple model. Okay? what I'm going to do here is I'm going to plot for rods the probability of seeing noise and the probability of seeing a single photon response near the visual threshold. So this is a photon absorbed per 10,000 rods. It's scaled to this level here. What our previous work showed is that the threshold is suited very close to this crossover point between the distributions, and it produces a distinction between the single photon responses and the dark noise. What the data that we've shown in GCAP knockout suggests to us instead is that despite the fact that these are wider and bigger, if this position of the threshold doesn't change, then it doesn't help you that much, because you're in this case where a lot of noise is getting over the threshold. Right, so even though you have better signal and noise ratio of the rods, if this isn't properly positioned, you lose your ability to discriminate single photon responses from noise in the bipolar cells. Now, interestingly, what this means, theoretically, is if we start to slide this to the right, to the ideal position, if this were ideally positioned with respect to this new distribution, you could put yourself into a position where you would get better discriminability of single photon responses from dark noise, right? Because you're starting with rods with a higher signal and noise ratio to begin with. So we tested this idea by applying a drug to our retinal slice that moves the threshold to the right. So this drug is for the glutamate receptor on the bipolar cells. And so we applied this drug, and what you see very clearly is that while the average single photon response is hard to see in a GCAP knockout, when you add just a little bit of this drug to slide the threshold to higher positions, now we clean this up and we get a much more reliable single photon response. So the next thing we'd do would be to shoot up this animal with the drug and see if it can see at lower light intensities, right? Well, as it turns out, we can't do that experiment because the drug is not specific. It'll target receptors throughout the animal. So we would need to find a way of having some kind of pharmacological or genetic manipulation that would affect only the bipolar cells and nothing else in the body. And indeed, we're trying different genetic ways of isolating where the threshold is set so that we can manipulate the expression of proteins that control it. I see this as going forward as one of the major goals of what we're trying to achieve. Now, with this in mind, what I wanted to convey to you is the idea that it's not just about having the most reliable single photon response in the rods, but if the circuitry isn't well poised to read out what the rods are saying, it'll never achieve the limits that are imposed at the level of individual rods, right? And on some level, this makes sense. You know, we assume that the entire retina is noiseless, but it's not. There are processes all over the place that are trying to pick out very small differences between signals and noise throughout the circuitry, and it's important for our ability to see near visual threshold. Now, let me end in the last 5 minutes or so by telling you about one future direction. So I told you about this threshold. The threshold is present in the rod bipolar cells. But what if we could actually change the amount of noise in the rod photoreceptors itself? What would that do? Well, as it turns out, there's a couple forms of noise in the photoreceptor cells. So once again, this is a suction electrode recording of current against time for a rod photoreceptor held in a suction electrode. Okay? And we're just going to record 60 seconds at a time with no flashes of light. Well, what you see is that there's really two forms of noise here. There's these periodic events called thermal events. As it turns out, if you took all of these and averaged them together, they have a size and shape that's identical to the single photon response. And we think that they're due to the thermal activation of that visual pigment molecule, which we call rhodopsin. And even though there's 10 to the 8th or 100 million rhodopsins per rod, they thermally activate very rarely to provide one of these events. But nonetheless, these are false positive signals from the rod that are going to occlude your ability to resolve other photon responses. But there's also this other form of noise that is happening all the time that's caused by fluctuations in a transmitter molecule inside the rod. And indeed, when we close all the channels in a rod, we see that that form of noise also goes away. So we've bred two strains of mice that have changes in these forms of noise. Now, I won't go through this, but to make a long story short, you can reduce the number of discrete noise events simply by looking at rhodopsin heterozygotes. or mice, rather than 2 copies of rhodopsin, only express 1 copy of rhodopsin. They have half the rhodopsin, generate half the thermal noise. On the flip side, we can reduce that continuous noise by reducing in half the concentration of the molecule that controls that internal transmitter. So you have half of this effector molecule, you have half the turnover rate of the transmitter. Okay? And these are just proof of principle that indeed we can reduce these noise sources. And as it turns out, we reduce them selectively without touching the other and without influencing the size or shape of the single photon response. So we're now in the process of evaluating in these mice how behavioral threshold is altered and how the signals through the retinal circuitry are influenced. So in terms of behavior, the water maze is actually quite painful. Okay? It takes a long time. The mice are swimming. They're doing all kinds of bodily things in the water. You know, it becomes a real pain in order to characterize threshold. And in fact, when the mice can't see the target, they're swimming around for 40 seconds or 30 seconds in cold water. So you don't get that many trials when you start getting close to threshold. So we've been designing another behavioral task that takes into account a mouse's ability to perform a learned behavior. So tasks like this have been used throughout visual processing. Now, here's an example from Matteo Carandini's lab of a mouse looking at a contrast grading. So the idea really simply is that they're primed by a black target. If they can see the contrast grading on one or the other side, they pop their head through a hole. And they break a beam, which registers as being correct, and they're given a reward. So you can see in this little 30-second clip, okay, didn't get it right. There's some punitive action that happens because they didn't get it right. Then they go back to doing this trial. And so these methods have actually been worked out quite well to do mouse behavior. And in fact, we've built a device essentially to do something very similar, where the mouse pops its head through the center, and there's 2 LEDs on the left or right that project a small spot on the retina. And essentially, by doing what we call a 2 alternative force choice task, we can now put ourselves in a position where we can discriminate very quickly where a mouse's visual threshold is, because there's a definitive statistical framework for evaluating these kinds of experiments. And indeed, since we've moved to UCLA from USC, we haven't reinitiated these experiments yet, but we're in the process of doing so this summer. So what I'd like to do is I'd just like to end by saying that the visual system, I think, ends up kind of getting the shaft when it comes to neuroscience more generally. Because the system is so robust, because we know so much about it, it's not appreciated how deeply we can delve into some of these research questions. Particularly research questions like this, where we're talking about the ability of circuitries to resolve single photon responses from noise and what it means for detection sensitivity. So what I've told you today is that the signal and noise ratio of the sensory receptor itself matters, but how it's processed as you go through circuitry matters too. And our lab is continuously trying to understand the mechanistic basis, not only for how signal photon responses go from rods to rod bipolar cells, but how they're relayed through the rest of the retina to affect the output cells of the retina. So the last thing I'd like to do is I'd like to acknowledge the people who've done the work. Haru Okawa was a graduate student in my lab that did all of the GCAP knockout work. He's now a postdoc with Rachel Wong at the University of Washington. The stuff on noise is done by postdoc Johan Palberg, who's sitting right there. The behavioral work was started by Dutch Ratliff, who's unfortunately not here today. I've had the pleasure to be supported for this work by the NEI and some private foundations. And let me just end now by saying that you guys have an incredibly difficult job. Faculty members are incredibly narcissistic. They don't follow deadlines. Am I right, Joe? Yeah, okay, I'm just joking. Yeah. But let me say this, despite the fact that many of my colleagues can be objectionable at times, I think we all appreciate the fact that at a top-tier research institution like UCLA, that the kind of support that you provide to us, dedicated people providing real support, makes it possible for us to do this. We can't do any of these experiments without your help, so thank you very much. So can I take any questions with the last couple minutes? Or tell you what, Why don't we wait a minute for anybody who would like to? Or am I so engaging that everybody wants to stay? Okay, fair enough. Yes?
How far are we away from a cure for night blindness?
It's surprisingly close. So the way I liken this is that in order to really understand how something works, you have to take it apart and understand mechanistically what's going on. So if you were to take a sports car into a garage and you were to ask me to, what's wrong with this, I couldn't tell you. You would have to, take it apart and look and see. I think with respect to night blindness, we now have a deeper insight about how to cure night blindness because we're understanding how the single photon response signal and noise is being regulated. So drugs like this that change the threshold position and other things might help you optimize if they can be delivered if they can be delivered in a very precise way and control function very locally. You might find that experiments like this give you insight into how to improve the signal-to-noise ratio of the rods, which will impact detection threshold. Yes?
The thermal noise, every time there's a photon receptor, does the thermal noise happen every time? Or is that...
So no, the thermal noise that I'm describing is happening in darkness.
Right.
Now molecules are not thermally stable forever, but rhodopsin is remarkable. Okay? There's 10 to the 8th rhodopsins in each photoreceptor outer segment, each rod outer segment. And on average, you see one of these thermal events every 60 seconds. That means on average, each molecule is thermally stable for approximately 1,000 years, or longer than its lifetime. So the thermal stability is actually remarkable, but you still see this thermal noise because there's so much rhodopsin packed into every rod. Yes?
So in your experiments, you were measuring basically the voltage on the cell with your electrodes. But essentially what transmits the signal, as you say, is a neurotransmitter which diffuses from one cell to another.
Yes.
Are there any microscopy techniques which would measure the neurotransmitter concentration directly?
The answer is there is starting to be. One of the most prominent examples in the retina is being used by a colleague of mine, John Dem at Yale. It's something called glue sniffer. It's something from Genelia Farms where essentially it's a glutamate receptor binding protein attached to a fluorophore. And if you've expressed that on the postsynaptic membrane, or the cell that's receiving the signal, that it changes its fluorescence. And so you can use the amount of fluorescence as a measure of how much glutamate is being released. But quite frankly, we've not come to the point where we can really robustly and quantitatively establish how much neurotransmitter is present, except that it's changing, and we can look at every point along the circuitry and see how it's affecting the voltage of the subsequent cell. So we don't know exactly, but we can infer what the action is based on what the downstream consequences are.
Thank you so much, Dr. Sam, for a
Thank you.
wonderful presentation.